So Dr. Kotzé is a South African immunologist. She's based in Stellenbosch. She has interests in innate immunity to tuberculosis. She focused mainly in a PhD on biomarkers for TB, for diagnosis. And then, but she looked at this also within the context of other co-infections like TB and HIV. Sorry, not TB, helminths and HIV. And then she has been in the same group looking at, for a postdoc, looking at myeloid suppressor cells. But she then recently moved more, or that project sort of evolved towards looking at more of a disease site and developing a 3D spheroid model for TB granuloma, which is quite interesting, I guess, in a way, because, as you know, there are many different models that are used for TB granuloma, and some of them a bit tricky. So I think having an in vitro system to exactly do that, and especially directly from patients, could be quite helpful in many ways. And I think that's what she's going to tell us about. But she also has other passions, also including teaching future scientists about multicolor flow cytometry. And also she enjoys sharing her experiences in science and immunology to the younger generation. So over to you, Leigh.
Thank you very much, Sabelo, for the introduction. It was very kind. Before I get going, I would like to give a big thank you to everyone who is involved in this project. Today I'll be presenting the work that I've done during my postdoc with the Immunology Research Group, and it was an absolutely massive collaboration. And it was very humbling and just a wonderful experience to have the expertise of so many peers, to putting their minds together to have a really nice collaborative project with a very, very satisfying outcome. So a very big thank you to everyone involved. So as it says, my postdoc project was largely based around the establishment of a patient-derived in vitro model for human tuberculosis granulomas. And for our model, we established we established it using a magnetic levitation-based method. And yes, while I get over the first of my jitters and technical issues, I'm going to talk very briefly about Mycobacterium tuberculosis. This is a pulmonary pathogen and is the causative agent for active TB disease. And until recently, it was the leading cause of death from a single infectious agent until we were unfortunate enough to have the SARS-CoV-2 pandemic. And this is a very interesting pathogen. It has eluded us for many, many decades and even centuries. And we see that the method or the mode of infection is not as simple as either infected or uninfected. We have patients who fit a continuum or a spectrum of infection, which goes from as complicated as an initial infection, which is cleared, all the way to active disease, where transmission is possible. But in between your cleared infection and your active disease, you have patients who present with latent infection, who are not contagious and manage to control infection without being able to fully clear it. And you also have complicated cases where people present with subclinical disease. And these two groups are really interesting because they serve as a reservoir in our populations and our high burden communities, which could allow for react or progression to active disease. And these people can therefore act as reservoirs for future disease. So infection with MTB, or Mycobacterium tuberculosis, we know is responsible for altering host cell responses by acting at an epigenetic, phenotypic, and morphological level, which is also known, just moving my face because I keep looking at myself and I see my eyes are dwelling towards the right of the screen. Sorry about that. So we know that the epigenetic changes are very much dependent on the spatial and anatomical orientation of the human immune cells within the site of disease. Traditional studies into MTB infection dynamics have been very much limited to primary blood cells, considering the relative ease of obtaining these kinds of samples from a patient. But we know as scientists that tissue-specific factors establish tissue-specific immune regulation and responses, creating niche microenvironments in every single corner of the body. For example, we know that a TB granuloma, the orientation and organization of specific immune subsets are incredibly important in determining the outcome of a granuloma's establishment, thereby determining whether or not they will be protective or non-protective in terms of their pathology. So we really need to regroup as a scientific community and place more attention on location-specific response dynamics in our experimental approaches, because realistically, how can we rely on data based on cells that have been retrieved from compartments of the body that are not the site of disease to make accurate inferences or assumptions on how the immune response is progressing during an infection? So with spatial and anatomical orientation in mind, we need to focus on investigating these cells, like I said in the previous slide, in a manner which more effectively represents or mimics the native kinetics of these cells and their organization for the disease and cell in question. So this is where the field of 3D cell culture methods become incredibly important and a very useful tool. Cells that are grown in 3D cultures are grown in 3 dimensions, and they demonstrate a closer proximity to one another with natural cell-to-cell attachments forming. And this allows for much more effective cell-to-cell communications because of the improved formation of gap junctions between the cells. And this allows for communication via ion exchange, small molecules, and even electrical currents. And we know that cells that are cultured in 3D have been shown to respond to mechanical cues, whereas those in 2D culture do not. In the cancer field specifically, where 3D cell cultures have really been well developed and taken off from, these models have revealed why some in vitro drugs or vaccine candidates that have yielded promising results in vivo preclinical trials go on to fail in subsequent in vivo models. So the problem then comes in that we have these 2D monolayer cultures or traditional cultures, as I'll go on from here, that have been used as a proxy for studying the site of disease, but these cultures consist of randomly arranged cells without any organization specific to the in vivo environment. And because of this, the monolayer cultures fail to recapitulate the key elements of the cells that reside in complex tissues, such as for example, a TB granuloma or even a tumor cell, or a tumor, sorry. So the question is then, what is the implication of this on clinical research in TB? As I mentioned in the first slide, TB disease is characterized by a formation of a granuloma structure within the lung at the site of infection. And the main purpose of the structure is to contain and hopefully eliminate the infection. Now, there's a lot of data that demonstrates that these granulomas are incredibly complex and their microenvironments rightly so. And these are not adequately represented by a 2D monolayer where the cells are cultured in a flat arrangement. If you think about a granuloma, it is a round ball-shaped aggregation. So we also know that granulomas are highly dynamic, and although their function is to hopefully eliminate but normally contain infection, the reality is that the granulomas could actually be protective or non-protective depending on the microenvironment of the host and the interplay with the invading bacteria. Therefore, by implication, the cellular and biochemical factors regulating granuloma formation, their maintenance and function, and also their resolution in humans is incredibly multifaceted. And this is where the field is in the process of identifying features specifically of host immunity that can tip the balance between restrictive and permissive granulomas. And this is where I believe the identification of true correlates of risk and protection are incredibly invaluable and needs to be elucidated for the assistance of our research into TB and ultimately the eradication of the disease. So the granuloma structures are incredibly dynamic. They're typically characterized by a core of alveolar macrophages, which are infected and uninfected. I forgot to put my pointer on, very sorry. So we have a core of macrophages that are uninfected and infected. And these recruit additional myeloid cells as well as lymphocyte lineages to the site of infection to form this protective cuff or barrier, as it were. And the fate of these granulomas is highly dependent on the balance between pro and anti-inflammatory regulators within the immediate microenvironment. And the implication of clinical research then is to define the dynamic balance between the regulators, because this ultimately determines the fate of pathology and bacterial control. So we generally have granulomas that remain resolved and do not allow for permissive transmission. But if there is a tip between the pro-inflammatory and anti-inflammatory mediators towards a non-protective phenotype, we have these unresolved granulomas which burst open and allow for the transmission of the bacteria to further sites of the lung. So we also have a complexity where we don't have one single type of granuloma form within the lung. We may have caseating granulomas, which are characteristic of active disease and transmission, but patients with active disease can also present with non-necrotizing granulomas, which have live bacteria but do not result in a caseum forming, which is typically the characteristic of transmission. In contrast, we have persons who have latent TB infection, and these people represent a group within the spectrum of TB infection that are characterized by also a continuum of more or less dormant lesions, which may also be healed. And typically you see a lot of fibrosis within these lesions, but it is not limited to only fibrosis. Then what complicates matters further is that any one individual who has active TB disease may present with multiple lesions, and not only multiple lesions, but these lesions may all be of different types. So this makes targeted treatments very, very difficult for our field. So in addition to the cell type composition of granulomas being incredibly important, there are also numerous publications that demonstrate the spatial positioning of these cells within the structure is greatly determinant of the protective outcome of individual granulomas. We know that the presence of T cells within the granuloma are highly important for granuloma containment, as demonstrated by Gila Kaplan, who showed that the extent of immunoactivity is determined specifically by the density of CD3 T cells, so all T cells within the macrophage-rich areas, which then determine the trajectory of the TB granuloma. Interestingly, in studies where T cells were identified within the central core of the granuloma, these structures could not adequately contain infection. So more recently, it was shown that in terms of homing dynamics of recruited T cells from lymph nodes, it is not the defective homing of the specific T cells to the granulomas, but rather defects in their positioning within the granuloma that poses a major barrier to T cell mediated immunity. And for example, a granuloma, granuloma CD4 T cells are found within the outer layer or outer cuff, and very few localized towards the center of the core. So in addition to these in vivo experiments, we also have a lot of systems biology approaches which show similar findings. And here they've demonstrated that the structural organization of the granulomas, as well as the recruitment of non-specific T cells, MCB non-specific, likely result in reduced responsiveness to treatment and also containment. So in this regard, spheroids have emerged as a very powerful tool to study host immunity, not only in the context of tumor biology where they were initially developed, but also in granulomatous diseases. And they have been shown to have huge translational potential between structures which are similarly designed. So tumor spheroid structures are much better at representing the in vivo tumor morphology. And they have shown with these studies that they have cell connectivity, polarity, and gene expression, and also the tissue architecture much better mimics the native tumor biology, making them more physiologically relevant and predictive compared to 2D models. Spheroids have been studied with different technologies, and these studies have shown that cell proliferation and viabilities differ in the various microenvironments depending on different tumor types, depending on the cancer types. The most obvious difference between the 2D and 3D culture systems are undoubtedly the architecture of the collection of cells. And again, the context of this provides a demonstration of the importance of the nature of cell-to-cell contact and the formation of extracellular matrices forming around these cells, and how important this is for the recapitulation of actual events happening within the host. So just a reminder, this 2D monolayer cell cultures do not allow for dynamic exposure, dynamic interactions rather, with one another, because they rely on uniform exposure to things such as drug molecules, and their exposure to oxygen and nutrient gradients are highly uniform. In comparison, significant work on the tumor spheroid models have shown that the spatial orientation, once again, is highly important in order for these spheroids to demonstrate tumor-like features. And they have shown to have necrotic and hypoxic cores with nutrient-deprived characteristics, altered energy availability, as well as quiescent cellular states. And these gradients gradually increase towards the outer edge of the tumor spheroids, where the microenvironment then returns to the homeostatic state of high energy and availability and an abundance of oxygen and nutrients. So this has led to an increased understanding of how host immunity works specifically in cancer, but this has a huge translational potential to structures that have very similar spatial and anatomical orientation bases, such as the human TB granuloma, which we know has a deficiency in oxygen within the central core that is then reestablished towards the outer edge, which very nicely mimics a tumor spheroid. So with this in mind, we have adapted a tumor spheroid model to assemble our own in vitro TB granuloma structure. And in order to do this, we have taken primary airway cells, or alveolar macrophages, from the lungs of consenting participants using a bronchoscopy. And we also collected autologous peripheral blood, from which we isolated T-cells. So the alveolar macrophages were used to construct the core of the granuloma, while the T-cells were used to construct the cuff. And for each granuloma, we included exact monolayer controls in that they had the same ratio and composition of cells, except for the structural and spatial organization. And then the main aim of this project was ideally method development, because this model had never been built before, and we wanted to ensure that the model will be viable for downstream processes such as flow cytometry, immunofluorescence staining, interrogation of gene expression using RNA-seq. We also wanted to investigate cytokine and chemokine expression, as well as bacterial control of these cultures, comparing our 3D structure versus our traditional culture in the 2D monolayer. And then the purpose of creating such a model would then be to use this as a mainstream platform in all laboratories to interrogate not only novel drug targets, but things like mechanisms for drug delivery, host-directed therapeutic targets, interrogation of correlates of risk and protection. And I mean, the list goes on for the kind of availability that this platform would provide. So just a quick description of how we actually generated the granuloma structures. We infected alveolar macrophages with MTB or BCG, and the non-internalized bacteria were washed away and then the structures were cultured overnight. We then added specialized nanospheres, which are polyol lysine coated gold iron oxide nanoparticles, which bind electrostatically to the surface of the cells. This allows for the macrophages to be magnetized, and after an overnight incubation, we apply an external magnetic force to the top of the culture plate. This allows for the levitation of the macrophages off the base of the culture plate to the air-liquid interface, and allows the macrophages to interact within a 3D microenvironment. We currently add the T cells at day 4 after post-infection, and at the day 4 time point, we remove the levitation drive and apply the external magnetic force to the base of the culture plate. This just brings the macrophages to the bottom of the well to allow for the interaction with the T cells that we have added. It's very important to note here that our T cells remain unlabeled. And this is because we wanted to rely on natural chemotactic gradients to allow for the T cells to migrate towards the core. We then culture the granulomas for a further 3 days up until the day 7 time point at this stage, after which we process them for downstream applications. So work performed by one of our master students, Caleb Peterson, with the help of Jurgen Kriel from the Central Analytical Facilities at Stellenbosch University, have confirmed that these nanoshuttle or nanospheres bind electrostatically to the surface of the cells. And work performed by Caleb last year also confirmed that these nanospheres do not activate the cells transiently, nor do they activate the T cells by bystander activation, because the T cells remain unlabeled with the nanospheres. So this is very promising and is a very nice tool to use. Using basic light microscopy and an inverted microscope, we can already begin to visualize the basic morphological differences of our structures from the 24-hour time point. And what we initially have is our core of alveolar macrophages, as described previously, and one can see that with the labeling of the nanospheres, we have this dark appearance of the macrophages. But this is not only because of the nanospheres, but also the loading of carbonaceous particles, and this I'll discuss a little bit later. Then at the day 5 to 7 time point post-infection, once the T cells have been added, the size of the structure increases quite significantly. And we can see that the lymphocytes begin to migrate towards the core and eventually begin penetrating the relatively outer cuff of the macrophage core. We have not yet seen, and hope not to see, any T cells within the central core of the macrophage, of the granuloma structure, sorry. And you can see here we have our macrophages that are quite large and our T cells that are busy migrating towards the core. We have also demonstrated using H&E staining the basic morphology of the cells at the core, which begin to take on a multinucleated giant cell. I'm very sorry about that, forgetting my words today. So we see these multinucleated giant cells beginning to form at the core from the macrophages that were initially used for the core formation. And then we have the T cells forming the cuff. We've also been able to perform ZN staining on the structures, both uninfected and infected, to identify acid-fast bacteria and where they are relative to the structure's size. We've also been able to image these structures with an ARIEScan 880 or LSM 880 ARIEScan confocal microscope. And what we do is we first embed our structures in OCT, which is an embedding media, and then we cryosection 7 micron thick sections, which are then used for staining. We stain with CD206 for alveolar macrophages and CD3 for our T cells. And our confocal images very much represent what we see in the light microscopy images, where the core is almost exclusively made up of macrophages and our T cells are localized towards the cuff. So this is very promising, as what we've seen in the non-human primate models is largely reflected by our granuloma structures. And this is promising because previously for granuloma studies in TB, we've had to rely on non-human primate models to study the response to infection at the granuloma site, because one can imagine how difficult it is to obtain an entire granuloma from a person who is very, very sick with active disease. Resections of their lung are just simply not possible. And then we have other issues like murine models not being absolutely representative of the human immune system, and only the Kramnik models really create this granulomatous disease upon infection as well. So this is very promising that our structure is reflective, of the non-human primate studies of granuloma models. So phenotypically, we have shown that our granulomas are reflective of non-human primate models. But we also wanted to address the immunological kinetics of these structures. What is happening at a kinetic level upon infection? So we did so by staining for necrosis, HNGB1. It is a damp molecule which is used as a proxy of DNA damage and necrosis. And typically, the HNGB1 protein is constitutively expressed within the nucleus of the cell. And then upon hypoxic or stress conditions, the HNGB1 molecule translocates to the cytoplasm. And what we found with our granulomas that are affected with MTB, the core cells of the granulomas showed the cytoplasmic translocation of HNGB1, indicating that necrosis had indeed begun to occur. And this was at the day 7 time point. Interestingly, the cells at the cuff of the granuloma structure did not display any necrosis beginning to occur, as can be seen by the expression of HNGB1 at the nucleus. And this pink color is because of the co-localization with the DAPI stain. What is very important from this image is that this feature, or this immunological response, happened really organically. And this really drives home the importance of using a 3D culture model versus a traditional 2D monolayer, because we did an optimization experiment whereby we tried to observe HNGB1 and necrosis occurring in a monolayer structure, also using macrophages, but this did not occur at all unless we mechanically induced an anaerobic environment. As we can see here, we saw no upregulation of HNGB1 in untreated culture conditions, but upon the induction of the anaerobic environment, we began to see the cytoplasmic translocation of HNGB1. We also investigated the marker HIF1 alpha as a marker for hypoxia, but unfortunately, this marker is relatively unstable at the slightest reintroduction of oxygen to any kind of system. So we are busy looking at alternative markers for the interrogation of hypoxia. We only managed to identify a slight upregulation of HIF1 alpha within the cytoplasm, but this is not indicative of hypoxia. HIF1 alpha basically acts in opposite mechanism to HNGB1, where it is constitutively expressed in the cytoplasm and then translocates to the nucleus upon hypoxia. So even though we saw an upregulation, there was no nuclear translocation, which speaks to the sensitivity of this marker in culture conditions. So I hope that this optimization truly endears you to the use of 3D culture models over 2D, because we could not achieve the necrosis factor without mechanically inducing an anaerobic environment in the monolayer condition. Just want to take a sip of water. Moving along from the immunological kinetics, we also wanted to see if the spheroid granulomas would form complex extracellular matrices, as reported for tumor spheroids. So we had available the marker for Phalloidin, which stands for cytoskeletal F-actin. And here we could see that in this, the Phalloidin is stained in red in this instance. These structures exhibit intricate cell-to-cell interactions and cell ECM interactions, at least after 7 days of culture. We also made use of a fluorescent MTB strain, and we identified that at the end of day 7 of post-infection, we found that bacilli were most abundant within the core, or at least with the cells localized at the core. However, we did see some extracellular bacteria located towards the cuff. These results show that we can detect MTB after 7 days of culture. In this case, this was not a protective granuloma, but we will have to look into the optimization of MTB infective doses going forward, considering the plan to extend culture periods to observe more later stage and mature granuloma formation in the in vitro setting. And I would just like to thank Caroline from the Immunology Research Group for all her help with this microscopy, as well as Dirk and Susan from the UCT Imaging Facility. A lot of optimization went on in the background to get to this point, and without them, we would not have been able to do this for sure. So going back to those carbonaceous particles I mentioned a few slides ago, when we work with patient-derived alveolar macrophages, it can be quite challenging, because in settings where burning of biofuels and smoking prevalence is very high, alveolar macrophages tend to internalize these carbonaceous particles, making them highly autofluorescent in nature, because the carbon particles irregularly scatter light, making the true fluorescent signals incredibly blurred. Previous work by my colleagues in the Immunology Research Group have nicely demonstrated by flow cytometry how it is relatively impossible to really sort out true signal from autofluorescent signal, particularly in samples which are visibly black coming out of the lung because of high smoking or biofuel burning prevalence. And we can see by light microscopy in a Rapidev stain how incredibly loaded these macrophages become with these carbonaceous particles. So because of this, we struggled to do some flow cytometry on our own granuloma models, but we managed to get around this by removing the macrophages from culture after the 7-day period. Luckily, going forward, we have acquired the Cytec Aurora, which is a spectral flow cytometer, and this allows us to unmix the sample data from the autofluorescent data, and has given us the ability to investigate the entire 3D granuloma structure in one go going forward, which is very, very exciting. And this is just a demonstration that Andrea Brichmead did from our lab, demonstrating using a conventional flow cytometer how one really struggles to obtain any true signal compared to the spectral flow cytometer after unmixing and subtraction of the autofluorescence signal. We can really nicely tease out the true fluorescence that is happening. So because we hadn't acquired the spectral flow cytometer at that point, we had to remove the macrophages from our granuloma structures and then run flow cytometry on the lymphocytes and other myeloid populations. We could perform viability checks for these granulomas, and it is very promising that after 7 days of culture, our 3D granulomas maintained very good viability, and this was nicely comparable to traditional monolayer cultures. And this we confirmed with both trypan blue, and then for the granuloma and the monolayer for aquaamine via flow cytometry. We managed to interrogate markers such as CD3, CD14, and CD56 for the interrogation of T cells, monocytes, and natural killer cells with our granuloma model. And I just want to point out here, we did have very few, data points for the pilot project. We had very few data points for our pilot project because this was a proof of concept, and just to see, like, that is this model viable for further development. But we are very happy from our flow cytometry results that our granulomas are capable of being used for flow cytometry without the compromise of cell surface receptor integrity, especially after such a long culture period and after the removal of the macrophages. We have also been able to quantify extracellular cytokines using a multiplex Luminex assay. And although we have a small donor size here, we managed to show that soluble mediators can be quantified in as little as 50 microliters of granuloma culture supernatant after the 7 days. Interestingly, we measured in the same assay for some of the analytes higher quantities of some cytokines produced by the same cells when in spheroid composition compared to those that are cultured in 2D culture. We of course have to look at many more data points to verify this, but it does point towards the possibility of cells in a 3D conformation behaving much different to the ones in the monolayer. We also managed to use the lyzed single cells to assess CFU burden or bacterial burden using CFU, sorry about that. And here we used the 4-hour post-infection time point, and we also measured CFU after 7 days of culture with the 3D granuloma compared to our traditional monolayers. And while we have to increase the data points available here, there is a trend for differential CFU between the two conditions, which is very promising, and also again demonstrates the difference between culturing in a 3-dimensional environment versus on a flat monolayer plate. So finally, the single cells obtained from our granulomas were used for bulk RNA-seq. We were able to extract very good quality RNA from the samples with the RNA yield ranging from 31 to 160 nanograms per mole per granuloma. I cannot infer any significance in immunological states here, with unfortunately having only 2 participants which we could use for the RNA-seq experiment. But I just want to mention that what we did find was that the results for one individual, shown here in green, separated very distinctly from the second participant granuloma from a second individual. These 2 individuals were both healthy donors of the same sex, and all other clinical criteria that were captured were identical. The only difference being that the one was a smoker and the one was not. For these 2 individuals, we found that 138 genes had significantly differential expression between the infected granuloma structures. And of these, 70 were upregulated in the granulomas from the non-smoker, whereas 68 genes were downregulated in the granulomas from the smoker. So this really surprisingly demonstrates the difference in immunological responses between someone who smokes and someone who does not. Looking at macrophages only, so the alveolar macrophages, we found that upon infection of the granulomas with MTB, more than 100 genes were upregulated in the 3D conformation. The top 10 upregulated genes represented pathways linking to organelles, linking organelles to the actin cytoskeleton for subcellular organization, proteins of the secretory vesicle machinery, intracellular signaling, components of T-cell engagement, and quite a few others. In comparison, we have the differential gene expression from the granulomas compared to traditional monolayer cultures. And here we found that those genes from our 3D granulomas upregulated about 640 more genes and downregulated 523 genes compared to the traditional culture. And I must just point out here as a reminder that our granuloma structures were identical in size, in cell source ratio and composition towards our traditional models. So both monolayer and granuloma culture had the same number of cells at the same ratios and the same types. So analysis of the top 10 differentially expressed genes for the difference between the 3D granuloma and our traditional culture showed that 3D granulomas augment the transcription of proteins related to inhibitory synapse development, neuropeptide receptors, zinc finger transcription factors, actin filament proteins, and T cell activation. And this suggests that the differential gene expression due to geometric organization and spatial organization of cells is definitely much larger than the differential gene expression observed for a monolayer, which again just highlights the importance of investigating immunological kinetics and cells within an environment that truly represents and mimics the natural cellular environment. And while this was only 2 participants, it does show promise and a lot of interest for future targets of validation and verification for this model. So in conclusion, I'm very happy to say that we established a patient-derived in vitro granuloma model using magnetic levitation. And this will definitely allow for the future interrogation of the host immune response to infection and the interplay between MTB and the host. And the model allows for multiple readouts to be performed, including flow, Luminix, RNA-seq, and microscopy. And the most promising thing is that the structures created using this model are viable. The cells are viable after long-term culture, but we do aim to increase the time in which we culture these cells. And we are hoping to use these results from this pilot study to build new projects aimed at validation and further development, with our end goal being to have our model used as a platform for the interrogation of crucial biological questions about MTB infection in the host. I would like to give a big thanks to my host, Nanita Duplessis, for having this brain baby and guiding me in this project. Without her, I would not have had as much success as we definitely did. But this also goes to everyone else listed on the acknowledgement slide. Without the team effort, there would be many tears more and a lot more time spent than what we did before we reached publication. And it was definitely very inspiring to work with these people who had such in-depth knowledge of different techniques, like Tracy specifically from the MRC with her help with RNA sequencing and teaching me that. And thank you to everyone who's done small optimizations that have contributed to this work, as well as the patients who gave consent and the sisters at the A5 ward at Tygerberg Hospital for assisting with recruitment. And with that, I hope I have not taken up too much of your time, but I do thank you for taking the time to have a listen. Thank you.
Cool. Thank you, Leigh. Questions? So I think Born would allow people to, actually didn't take much of our time. We still have plenty of time for questions. So hopefully Born would be able to unmute some people. I think
yes, they can unmute themselves now.
Okay, you can. So Lisa, I think you can unmute yourself and ask a question.
Hi, thank you, Leigh.
Hi, Lisa.
Great presentation, and it's a really interesting model. I have 2 technical questions. One is whether you tried to use cryopreserved cells, both BAL and PBMCs. And the second question is whether you tried to add the BAL fluid to the cell culture as well, and what is the effect of that, and if there is, you know, any merit in exploring that if you didn't. Thanks.
Thank you for the questions, Elisa. Interestingly enough, all of our, for the majority of our granulomas, all of the granulomas were made using cryopreserved both BAL and PBMCs. So with the nature of the timeline for creating the granulomas, we receive BAL very opportunistically. So it's very difficult to set up a granuloma experiment with a day's notice. So we had to cryopreserve all opportunistic samples at that point and then begin the granuloma building at a later stage. We have not added the bowel fluid to the structures as of yet. The reason for this being, let me get my bearings quickly. I think with a lot of the granulomas from smokers, we had a lot of contamination of carbon particles within the fluid, which we couldn't quite accurately filter out, even though they weren't internalized within the macrophages. So we didn't want to further complicate the kinetics of what was happening in culture by adding more carbon to the system. But it definitely would be very interesting to do so. I think there would be a lot of growth factors at play that we may be losing by filtering out or not adding bowel fluid, but we would need to assess their levels, I guess, to determine what concentration of fluid to add or not to. And I do see Nolita has responded. I agree. I think the saline would definitely influence the cell culture model, because bowel fluid is essentially a saline retrieval of cells from the lungs.
Okay, and Rachel?
Yes, Rito speaking. Can you hear me?
Yes.
It's a very interesting talk. Thank you. I'm wondering of the marker that you use to stain these alveolar macrophages, CD206, which is a mannose receptor, which is a marker for alternative activated macrophages. You seem to have very strong staining for these M2 macrophages. And would that really resemble the NHP granulomas as you as well tried the M1 markers? And the other question is just for the RNA-seq. I saw in detail that you actually stimulate them with Mycobacterium bovis. But what was the rationale of using M. bovis and not rather MTB or the strain H37RV that you have used before to infect these alveolar macrophages? Because certainly the transcriptional profile, it changes between M. bovis and MTB.
I think, let me begin with the first question. That was regarding the CD206 marker. Yes, there was quite a strong staining. Let me just think about this. So there are definitely a lot of alternative markers to be used for macrophages, and I think the jury is out really on what is truly a M2, M1 marker without also interrogating the functional markers involved with those surface markers stained. Because we had this proof of concept that we wanted to establish this model, we decided to simplify the staining as much as possible, because not only did we encounter quite a bit of autofluorescence with the flow cytometry, we also encountered some trouble with autofluorescence using the confocal microscopy. So we were really limited by the number of markers we could interrogate, and we had CD206 readily available within the lab. So we decided to go ahead with that, and it was on a color which we could use lambda unmixing to reduce the sample, to reduce the autofluorescence signal. So it worked out nicely that way. But we do have a much larger panel that we have established for our next round of experiments, which has included different M1, M2 macrophages, but also the phenotypic type markers to look at, like interleukin-4, IL-10. In terms of the RNA-seq data, we used, yes, we did use M. bovis to infect. The two that we used for the RNA-seq analysis was BCG, and we used this because of the dynamics. How do I phrase this? It was just much easier to use BCG in the BSL-2 environment and not have to worry about culturing these granulomas in the BSL-3 environment. BSL-3 has a lot of traffic at the moment, and the nature of the magnetic levitation of the granuloma model is such that it is very sensitive to fluctuations in temperature, and a lot of people bump the incubator. So we decided to use a BSL-2 organism so that we may monitor much more strictly the kind of foot traffic we have in the tissue culture. But it's definitely something that needs to be considered going forward. Now that we have established the model and we have confidently assessed the viability of actually performing RNA-seq on the model, we have started with H37RV infections, because the transcriptional nature would definitely be very different using different strains.
All right, thank you very much. Responses.
Jackson, Chopin.
Hi, thanks, Sabelo. Hi, thanks, Leigh. What a beautiful talk, beautiful work. I might have missed this because I joined a little late, but was the capacity of this model for you to track formation of giant cells or multinucleated giant cells? And I also wanted to check whether, as we know, that a very fundamental feature of granulomas in humans is caseation or necrotizing caseums and stuff. Is there capacity of this model to enable us to get close to that kind of cell death, for instance? Thank you.
Indeed. I'm going to begin with your second question, Jackson. So at the moment, the model is very much a snapshot of an immature early stage infection. With 7 days of culture, you can imagine after being infected in the lung after 7 days, you wouldn't readily have caseation forming within the lung at that point. So we are very pleased that we could identify at least HMGB1 in terms of an necrotic marker after 7 days. But we do need to optimize going forward, increasing the length of culture, because it is our hopes that we can investigate more mature and maybe later stage infection type models, whereby we can identify areas or regions of caseation, such as what you would see in the human at later stages of infection. And then capacity to track multinucleated giant cells. I mean, we can morphologically identify them after 7 days of infection, and we have cytospins that we make of each and every bowel sample that comes in prior to cryopreservation and use in the granuloma model, whereby we can identify kind of using a differential cell count what ratio of multinucleated giant cells we would have prior to granuloma construction. So there is capacity to track, you know, the kind of ratio that we're experiencing throughout culture for a time course. It's just, I don't think we got down to that.
Thank you.
Thanks.
Cool. And Mosem was asking whether the model just contains macrophages and T cells only.
Yes and no. So in terms of the autologous cells isolated from the blood, yes, they're only T cells. We isolate T cells using max speed isolation. For the alveolar macrophage core, we use adherence to specifically remove, isolate macrophages. So this is not necessarily only alveolar macrophages. You may also find some early monocyte lineage cells that would adhere to tissue culture plates. So we would expect to see those. So you also would have, in addition to your T cells, I guess we didn't exclude for cells such as natural killer T cells. So you would expect to find mostly T cell and macrophage lineages. But yeah, at this point in time, just for the method development, we wanted to focus on those two cells specifically. But our current experimental pipeline does include adding all cells from, say, the periphery. So we would have T cells, B cells, myeloid-derived suppressor cells, monocytes as well, with the end goal being to identify, if you had to not include a specific cellular subset, what effect would that have on the outcome of the granuloma formation and also bacterial control?
Cool. I probably just have one last question. So does the disease, or wherever you get the alveolar macrophages from, does that sort of influence the granuloma sort of formation, or would that influence your model?
Do you mean, say, the load?
Say because obviously the people that you're only gonna get the BAL from are likely to be people that are probably at a very advanced stage of TB. So is that in a way influencing the model itself, or it doesn't affect it at all?
I would expect it to. For the purposes of the model development, we use BAL cells from healthy donors only. So we would expect there to be activation of the cells that you would retrieve from a person with active disease compared to someone who was relatively healthy. So that would definitely influence the model, and the infection dynamics there would then also be different if we had to re-challenge with either BCG or H37. So that is definitely one of the next steps for us. And I think also it would also be interesting to identify which lobe the macrophages had to be retrieved from in relation to the site of disease in the case of those who are ill. What kind of bystander activation we would observe in those cells and how that would influence the model. So these are all many questions we want to ask with this model now that we know that it works.
Yeah, and does the BAL itself, as a very tricky thing to get, a limitation to the model?
Yes. So while a bronchoscopy is not a dangerous procedure, it's relatively safe, and we've had volunteers from our own lab who have also had bronchoscopies performed on them. Oh no, sorry, no. It is a relatively safe procedure, but participants are less eager to undergo such a procedure because it does take a lot of time out of their day. We have clinicians and nursing staff on hand who do a full clinical workup before the procedure, and then they are observed after the procedure. So it does take a lot of time out of the day, which is why they, I think, are less eager to volunteer or consent to it. So that is a limitation. We are also having a look at using monocyte-derived macrophages and trying and establish what kind of differences would be expected in the model between using alveolar macrophages and monocyte-derived macrophages. So it is a small limitation.
Yeah, and does the model differ substantially, say, from the air-liquid interface cultures, for example? I mean, would you expect something completely different, I suppose?
Sorry, could you maybe rephrase that?
So I'm just thinking around, so some lung models use, I mean, it's still kind of lower lung cells, but not specifically macrophages. And you can also sort of grow those cells in a 3D culture. But I'm just wondering whether that would be, maybe a different substitution to sort of doing a bronchoscopy, because that you can even develop it from, you can develop from nasal epithelial cells and things like that.
For sure. I think the capacity to use different types of cells to establish the granuloma is definitely there. You would just have to decide on which subset you want to label with these nanospheres in order to magnetize them to create the core. So I think there definitely is capacity to use nasally derived epithelioid cells, perhaps using even swabs, but I'm not entirely sure how we would isolate the cells from that. But yeah, I think it definitely could work.
Cool. I think, do we have any burning questions, last questions that people do want to ask, because while we have Leigh here, she can answer all those questions for you. Yeah, it seems like you have, I think they thoroughly enjoyed the talk, and you probably answered all the questions that they might have had. And thank you very much. I think that was very fascinating, and I think it's good to see that very complex things like a granuloma, which are obviously very difficult to get in humans and in other models that you mentioned, and that it can actually be done in a simpler method, and that's more accessible. Yeah, thank you.
Thank you very much. And thank you for the opportunity. Appreciate it.
Thanks.